screen share. Yeah, once I'm in screen share, I can't see the recording button, so I don't know if it's going or not. I just uploaded the video. It's still pending, so it won't be shown for the first meeting. I'll close that down. But I did get it going. It takes a while for it to convert. And I noticed like 3 minutes before starting class that it would finish. So I said, well, that might be enough time to upload it. And it just barely was. All right, any questions? If not, I believe we were on this slide. We finished that slide talking about the kingdom Animalia. Is anyone new? Should I talk about where we are? Well, it doesn't seem like anyone is saying anything, so let's go ahead and, excuse me. Let's go ahead and get started. Don't know what brought that on. So, microbiology and microorganisms. We're going to talk about that next. What comes under the heading of what is a microorganism? Maybe I can put that at the very top. There we go. I can blow that up. Nope, can't blow it up to that. So what is a microorganism? What comes under the umbrella of a microorganism? Well, we've discussed that bacteria and archaea, prokaryotic cells are under the realm of microorganism. And I think we also discussed that viruses and prions fall under the realm of a microorganism. But you should realize that these two are special, and that is they're not organisms. They do not have cells. You can argue about whether they're alive or not, although prions are, nobody argues they're alive, but they don't have cells, so they're a special microorganism and they're not an organism, but they do cause human disease, and that's why we study them and why I guess they're grouped under microorganisms. Certain of the eukarya are also under the umbrella of a microorganism. The protozoa, which do cause human disease, at least some of them. The molds and the yeast are also in this classification, falling under the realm of a microorganism, because to see the cells, even of the molds, you have to have a microscope. Some of the yeast, mainly, can cause human disease. Some of the algae also are a microorganism, the single-celled algae. None of these cause human disease. And then the helminths, the worms, fall under the umbrella of a microorganism. And they aren't microscopic. They are studied by microbiologists for two reasons. First, they do cause disease in humans. And the infective stage of the helminths are microscopic, meaning usually an egg of a helminth is the infective stage. All right, any questions about that? Viruses do fall under the category of a microorganism, even though they're not organisms, because they're acellular. Viruses do have either DNA or RNA in their core, and you could say that is the genome of the virus, but they do not have both DNA and RNA. It's either DNA or it is RNA. The core is surrounded by a protein, and it's called the protein coat. And that's either the genome of the virus, either DNA or RNA. It's surrounded by a protein coat. The coat may be enclosed in a lipid. So some viruses have a lipid layer outside of their protein coat, and we call that the envelope of a virus. Now, not all viruses have envelopes, only some of them do. And we'll talk about that later in the term. And when do viruses replicate? Does anybody know? Can they replicate on their own? Nobody wants to take a guess? All right. Viruses only replicate when they've infected a host cell. So viruses need a cell from another organism. Well, viruses aren't organisms. From an organism to replicate. They are not part of the three-domain system because they are not cellular. They're acellular. Any questions about that? They're really tiny viruses. They're smaller than prokaryotic cells. Roughly speaking, they're about 1/10 the size of a prokaryotic cell, and that's very rough. The largest viruses are approaching the size of the smallest bacteria. And here we're looking at small red spheres are human immunodeficiency virus in a red blood cell. So that's bad for the patient. So as we mentioned, there are three domains of cellular organisms: the bacteria, the archaea, and the eukarya. Let's do a little bit of a comparison. The bacteria and the archaea have the cell type prokaryotic cells. The eukarya have eukaryotic cells. When we look at their chromosome, generally the bacteria and the archaea have one chromosome, and it says the eukarya have one to many. Almost all eukaryotes have many chromosomes. I don't know of any that have one, but there might be one species that has one. Or one, I should say one family. It's probably more than one species, but most of the eukarya have many chromosomes. When we look at the bacteria of chromosome, it is one circular chromosome, and by circular we mean it's in a circle, and where it begins is where it ends. So there really is no one place where it begins and one place where it ends. And the chromosome has no histone proteins on it, and there's no introns. An intron is a region of the gene that does not code for protein. And that's sort of complex. Let's take a look at the eukarya. As I stated, most of the eukarya have many chromosomes, but apparently there's a few or one that has one chromosome. Those chromosomes are not circular. They are linear. If you look at the X chromosome, it has a beginning point and an ending point. The chromosomes of eukaryotes are wrapped around histone proteins, so they do have histones. And then the genes have all the genes of a eukarya have an intron in the gene, and that's a portion of the gene that does not code for the protein. Okay? The archaea are something sort of in between. They have a chromosome that's one, generally. It's a circular chromosome. It does have histones and it does have introns. And we won't talk about the archaea chromosome, so I won't ask you about that. I might ask you about the ribosomes of bacteria and archaea. They are a smaller ribosome than the ribosome of the eukarya, and we call the ribosomes of bacteria and archaea a 70S ribosome. That's the way biochemists measure ribosomes. And what it means is that is where the ribosome sediments in an ultracentrifuge gradient. And if you don't know what that is, we'll just say it's a characteristic of the size. And the eukaryotes have a larger ribosome, it's ADS, and it sediments deeper in the ultracentrifuge gradient. All right, any questions about that? If we look at the cell wall, the bacteria have peptidoglycan. You might remember my talking about that. And the archaea and the eukaryotes have no peptidoglycan in their cell wall. Of course, the eukaryotes, like animals, don't even have a cell wall. All right, any question about that? We can now compare the cellular types with the acellular microbes. And I'm going to be very brief on this because we'll talk about viruses and prions much later in the lab. I don't want to have much focus about that. The cell type of the cellular living forms is they're either prokaryotes or eukaryotes, meaning the cell has a plasma membrane, the cytoplasm, chromosome, enzymes, and stuff like that. The cell is complex. Viruses are acellular. They may have a genome. I would hardly call it a chromosome, but it's simple, the container of the viruses. So that's all you need to know about that. They don't have a cell membrane, no cytoplasm, but they do have a packaging. Prions are acellular. Essentially what they are is a single protein molecule. They don't even have a genome, so they're very simple. The chromosome of the cellular living forms, it's made of DNA. For viruses, the chromosome or their genome is either DNA or RNA, but it is not both. It's one or the other. And prions don't even have a genome. The cellular living life forms have ribosomes. That's the site of protein synthesis. Viruses and prions don't have ribosomes. And then the cellular types produce ATP, and the viruses and the prions do not. Okay, any questions about that? There's very little metabolism with the viruses, essentially, well, very little with the prions, and a ton of metabolism in the cellular life forms. All right, any question about that? Any question about anything we've discussed so far? Because we're about to switch on to a new topic. No questions? Then let's talk about the brief history of microbiology. The history of microbiology largely concerns, at least the early history, the debate over whether microorganisms could spontaneously generate or whether they came about from a previous microorganism. And we'll talk about that. The early days of the history of microbiology was known as the Golden Age of Microbiology, when microbiologists would look under the microscope and they would learn something new. And that was in the late 19th century and early 20th century, from about the years 1857 to 1914. And I'm not going to ask you to memorize these dates, okay? But early in microbiology, it was called the Golden Age of Microbiology. And it was just golden because whenever you were looking at something, you discovered something new. And this slide you can't see, but it's in your textbook. It's showing you the different discoveries that were made in the history of microbiology. And even when I've got it expanded like this, I can't read it. The Golden Age is right here, and we'll talk about that. Let's skim that. All right. So in the history of microbiology, oops, went too far. I'll get it. There we go. The history of microbiology goes back to ancient times, greater than 3.5 billion years ago. And this is the one date you do need to know. Over 3.5 billion years ago from today, we can see fossil records of the ancestors of bacteria in rock. And here you can actually see the picture, which it really doesn't show much, but I'll get it a little bigger so you can see that better. A fossil record of fossil of a bacteria from greater than 3.5 billion years ago. And there's more. And this is the artist's rendition of what they think it looks like. Okay? And why that's important is that that's the first evidence we have of life on the planet Earth. In the fossil record, a little over 3.5 billion years ago. The ancestors of bacteria today. Any question about that? Now, in prehistoric times, we have the first record of microbiology in action. The Babylonians had no idea about microorganisms and knew nothing about microbiology, but they wrote clay tablets down about different recipes for making beer. And that was 4,300 years BC. You don't need to know that date, okay? But that was an early record pertaining to microbiology. And then a little bit after that, 600 AD, the Mayans wrote records of fermenting a beverage from cacao, which we would call chocolate. They also didn't know anything about microbiology, but they wrote a record on fermenting chocolate. And those are two early records of microbiology in action. And it was the microorganisms that fermented probably, actually, I don't know what you ferment, some grain into making beer and fermenting the cacao to make chocolate. The first observations were made after Janssen developed the compound microscope. You don't need to know this date, but you do need to know the relative order of the discoveries. And I'll talk a little bit more about that when I end this slide. So in 1590, Janssen developed the compound microscope. And you don't need to know the date, but you have to know that the compound microscope was made before the first person used the microscope to look at living things and discovered in plants little boxes that he called cells. And that was Robert Hooke who did that. He took, I think the first thing he observed was the bark of the cork tree, and he did small shavings of it, and he saw these cells in the cork. And where the name cell comes from is when you look at them under a microscope. They're lined up in square rows, and that reminded him of the cells in a monastery. That's where the monks lived in the monastery. And that's where the term cell comes from. That marked the beginning of the cell theory. The cell theory wasn't developed until much later. But you had to see cells before you could obviously come up with the cell theory. The cell theory is that all living things are composed of cells. It was developed by others, like Virchow was the first person who wrote it up, but it came up much later than 1665 because when microbiologists were looking at animal cells, they could not see cells, at least the early microbiologists, in animal tissue, but they could see them in plant tissue. Does anyone want to guess why the early microbiologists could see it in plants, but not in animals? It has to do with the fact that the early microscopes were not that good, and they weren't that powerful, and they weren't that sharp of focus, and they're kind of blurry.
Because plant cells are bigger?
That would be a good reason if it was true, but it's not really true. That's not the reason why they could see cells in plants, but not in animals. That is a good reason, but it's not the reason. What is in cells that you can see and animal cells lack them?
Cell wall.
Yeah, the cell wall is why the early microbiologists could see cells in plants. It gave a clear demarcation of the cell. The cell wall is thick and rigid, and animal cells don't have that. All they have is a cell membrane. And the early microscopes weren't good enough to see the cell membranes. All right, the above is not specifically microbiology, although they had to be developed in order for microbiology to happen. And obviously you had to discover the compound microscope before anyone could use the microscope to see cells. And then you had to see cells before you developed the cell theory. Okay, any question about that? So that's why you need to kind of know the order of things. Does that make sense? All right, so you need to know the order things are done because you can't see cells until after you develop the compound microscope. So in the history of microbiology, it was largely concerning the hypothesis about how life originates. You don't need to know the years here, but in the 16 to 1800s, there were two competing hypotheses to explain how life began. And they initially did it with all living life forms. And then later, they only did it to microorganisms, meaning they used the two hypotheses to explain how life came about. The first was that life could spontaneously generate. Life could arise from inanimate matter. A vital force came into the inanimate matter and converted it spontaneously into a living form. And that's the theory of spontaneous generation. Any questions about that? The other theory is the theory of biogenesis, and that is that all living life forms arise from another life form. Like a baby is born because of what the parents did, and the baby came from the parents. Okay? Meaning the baby arose from a pre-existing life form, the baby's parents. Now, over a century ago, beliefs still included things like newts could arise from a log thrown into a fire. And when somebody threw a log into the fire and a newt crawled out, they'd say, spontaneous generation of the newt. And that is that whatever, the wood of the log and the fire could spontaneously create the newt. And that was the spontaneous generation of the newt. I suppose it was logical, but not scientific. Flies could emerge from manure. If you had your manure and you left it out, you would see that eventually flies would emerge out of it and then fly away from the manure. Maggots would arise in decaying corpses, and all of these things people said were spontaneous generation. You have a corpse, the vital force comes into it and changes it, and then maggots arise in the corpse. Some of them were amusing, none of them were logical. None of them were scientific. Nobody did any scientific studies on it for a while. Most of them were somewhat logical. The amusing one was somebody wrote up that if you put sweaty underwear in a jar with some wheat, grains of wheat in it, it would give rise spontaneously to the generation of mice. What happened was the mice were wanting the wheat in the jar, and they went in the jar to get the wheat, and then they got stuck in the jar, and that's where they really came from. All right, we're going to skip that. The first experiment answering the question about did life spontaneously generate, or did it arise from a pre-existing life form, came about from Francesco Redi. He did the first documented controlled scientific experiment. So he's important not only on answering the question about spontaneous generation of life, but he's also important because he gave the first controlled scientific experiment. And what he did was he had 3 jars that were uncovered, and he put meat in them. And then he had 3 jars that he completely sealed. And people said the vital forces couldn't get in the completely sealed jars, so that wasn't fair. And then he got 3 jars that he covered with a fine wire mesh net, and that way the air could get in and the vital forces could get in, but the flies couldn't get in. And he didn't know much about the flies, but he discovered that when he did that, the jars completely sealed, or the jars covered with the fine wire mesh, had no maggots in the meat. But the 3 open jars developed maggots. And so therefore, his experiment showed, or gave the first support, that spontaneous generation could not occur. That maggots did not arise from a vital force getting into the meat and creating the maggots. That the maggots had to come from some other place. And I don't think he discovered it. I think somebody else discovered that the maggots came from the flies laying eggs in the meat. All right, any question about that? Now, from this experiment on, people, meaning scientists, came to the conclusion that the larger life forms, meaning the multicellular animals and plants, did not spontaneously generate, that they had to come from pre-existing life forms. But that wasn't true for microorganisms. A lot of people, even after Francesco Redi's experiment, said that microorganisms, the small microorganisms, could spontaneously generate. And the reason why they did that is if you had a tube of media, I don't know how clear that is, let me blow it up. If you had a clear tube of media and then you left it out to the air, or even if you covered it and you let it incubate for about a day, it became cloudy with microorganisms. And so people said the microorganisms are spontaneously generated. Now what happened was that if you leave the tube open, microorganisms from the air will come in. And then the tube wasn't sterile. They put the sterile media in. Actually, early on, the media wasn't even sterile, but the tube wasn't sterile. And so, of course, microorganisms grew in the tube. Any questions about that? Before we answer the question about could microorganisms spontaneously generate, and we need to talk about microorganisms, and that is the first observation of a microorganism happened in the late 1600s to the early 1700s, meaning the 17th and the 18th centuries. And the first microbes were observed by this man here, Antony van Leeuwenhoek. And it's a Dutch name, so I could be butchering it, but I think I've got at least an English translation of it. He described microorganisms from practically every place he looked. He took scrapings of his teeth. He took observations of rainwater after the rainwater he collected for a day and then looked at it. He took scrapings from peppercorn infusions, and he looked at water in the river Thames, the river going through London. And he found microorganisms everywhere. Why he was the first person to see microorganisms was that he made his own microscopes. Let me shrink that just a little bit. And that's one of his microscopes. He was the best microscope builder and the best lens grinder of his day. Now, his microscope is not a compound microscope, meaning it has two lenses to magnify. His microscopes only had one lens to magnify. So in reality, it was a super duper magnifying glass or magnifying lens. And he would actually stick the specimen he wanted to observe. Let me show this. On that screw right there. And then he could adjust the screw, moving it up and down with that thing there. All right, any question about his microscope? He saw the first prokaryotic cells and the first protists, which were the first microorganisms seen. And he found these cells here, which I know you guys haven't really looked at bacteria yet, but that is very much so a bacillus or a a bacillus or a rod. And the same with these here and that one there. And he found spirochetes, which he's got drawn under figure G. And then figure E, which is really small to see. He found these here and he draw them. And these cells were so small that they were not seen for another 100 years. The reason being is the microscopes got slowly better, and over time, people started making better and better microscopes that finally were good enough to see the bacteria cells that Antony van Leeuwenhoek first saw about 100 years before everyone else. So that explains how good of a microscope builder he was. He saw bacteria 100 years, or about 100 years before anyone else. Any question about that? No questions? All right. Anything with the red, I'm not going to hold you to. This slide was saying that Virchow in 1858, this is like almost 200 years after people first came up with the theory of biogenesis, he actually wrote it up, and you don't need to know that because I'm not going to go into that. But I do want to talk about who's the person who came up with an experiment that finally answered the question, do microorganisms spontaneously generate, or do they arise from pre-existing cells? And the person who answered that was Louis Pasteur. In 1861, he finally disproved the hypothesis of spontaneous generation and confirmed the hypothesis of biogenesis, that life comes from pre-existing life. Before we go into that, we have to say that Louis Pasteur showed that microorganisms are in the air. He also showed that microorganisms can be destroyed by heat. And so you could sterilize the media with heat, and you could sterilize the tube with heat. And if you then made it so that the microorganisms didn't get inside the tube, but the vital forces could, you then could show that spontaneous generation did not happen. What he did was he had his flask like this, a long neck, put in the media, and then he bent the flask, you heat it, and then bend it like that. And then you have to heat the media to sterilize it. And then you just leave it open to the air. The vital forces can come in from the air and get into the media, but the bacteria from the air will settle in this portion of the flask, that S loop. And because of gravity, the bacteria don't go up the loop. All right? And he would show that if you took the media and you spilled it in here and then brought it back in here, you would get growth in this part of the flask. Not here because you were heating it, but in this part of the flask. So his S-shaped flask were used to disprove the theory of spontaneous generation. Any question about that? All right. So beginning with Pasteur's work, disproving the theory of spontaneous generation, the golden age of microbiology then really advanced. There were just lots of discoveries, like the discovery of the relationship between microbes and disease. That relationship is known as the germ theory of disease, that some diseases could be caused by microorganism, and we use germ for a microorganism that causes disease. Also, out of Pasteur's work came the idea or the concept of immunity, that we have an immune system that can make us immune to a disease. And also coming out of Pasteur's work was the development of antimicrobial drugs, a drug that could cure a disease caused by a microorganism. So a bunch of things happened. In 1850, Louis Pasteur developed the basic idea of the aseptic technique. And if you want to grow up a specific species of microorganism, you have to sterilize the container, the media, and then you only inoculate one species into that sterile media. And you inoculate it aseptically so that microorganisms from the air or the counter or the loop that you're using, the tool you're using to inoculate it, aren't contaminated. In 1881, like I said, you don't need to know the dates. I'm just saying them because of the way they are developed. Dr. Koch developed the technique for streaking for isolation on a solid media. Louis Pasteur actually did develop a technique for getting a pure culture using a liquid culture, and you use limiting dilution, and we'll talk about that in the lab. it was very difficult and time consuming to get a pure culture using Louis Pasteur's limiting dilution of a liquid culture. And then most of the cultures you set up were no good, only a certain dilution would work. Dr. Koch developed an easy way of getting a pure culture, and that is you just streak it on a solid media, and while you're streaking, as you're streaking along, I don't know if you can see what I'm doing here, as you're streaking along, you'll put down individual cells, and someplace on the plate, the individual cells will be spaced out enough that they'll form an individual and isolated colony. And if you take from that isolated colony, there's a good chance that it'll be a pure culture. And what we do to make sure we have a pure culture is we take from an isolated colony, streak it out a second time, and then when it is two isolated colonies, meaning it went through the first isolated colony and you streaked it out for isolated colonies a second time, if you take from that colony from the second time, it's a really high chance that it'll be a pure culture. It's a greater than a 95% chance, and that's what we do when we're trying to get a pure culture. Shortly after, Dr. Koch developed the technique for streaking for colony isolation on a solid media. Assistant of his who worked in his lab developed nutrient agar, which was a very good media for growing microbes, and it's also very simple and grew a lot of microbes, and that we call nutrient agar. If we can scroll down just a little bit. And about that time is the time when Sergei Winogradsky developed the idea that microorganisms are involved in the biogeochemical cycles, such as the nitrogen cycle or the sulfur cycle. That microorganisms are helping those elements cycle about in the ecosystem. All right. Fermentation and pasteurization came from the work by Louis Pasteur. That's why it's called pasteurization. And he showed that microbes are responsible for fermentation. He actually did this work to help out the beer and wine industry. Their beer or wine were becoming sour, and he found out it was microbial contamination, and it was converting the alcohol in the beer and wine into acetic acid, making vinegar, and it didn't make the beer or the wine very tasty. And he found out that microorganisms were also converting the sugar to make alcohol by alcoholic fermentation. So you need microbiological growth to make alcoholic fermentation. Louis Pasteur showed that microorganisms could also spoil food such as beer and wine. And that's true for other food too. And he discovered alcoholic fermentation and that the spoilage happened because of microorganisms. And then he came up with a technique for preventing the spoilage of the microorganism. And that is you heat the beer or the wine at a temperature not hot enough to evaporate the alcohol, but hot enough to kill the spoilage bacteria. And then your beer or the wine would last longer. And this is called pasteurization after Louis Pasteur. And it just makes he was doing it on first beer and wine to keep it from spoiling for a while. It will eventually spoil because the pasteurization doesn't make the item pasteurized, sterile, but it gets rid of the human pathogens and it prevents the item from spoiling right away. With pasteurized milk, for example, you can keep it for about a week after you buy it in the store and open it. And it's just using a high heat for a short period of time, and it doesn't sterilize the item like the milk or the beer or the wine, but it gets rid of most of the bacteria, and it gets rid of the bacteria which are human pathogens. It's used in a wide variety of foods. Today, it's mostly used in the milk industry or the dairy industry, but you can buy pasteurized beer. I'm trying to remember. Most beer is pasteurized today, like Cures, and I don't remember the other type of beer, sorry. The only beer that I know of that is not pasteurized, it's made in Colorado. What the heck is the name of them? I'm drawing a blank here. Maybe somebody can, in the class, can say, what is the beer that they filter and it tastes a little bit different than pasteurized beer? Anyone know? It comes from Colorado. Nobody knows, okay.
Coors beer?
That might be it. You know, I should know this, but I'm not a beer drinker, and I'm drawing a blank here. It might be Coors beer. So what's another type of beer? If Coors is not pasteurized, it's filtered, almost all the other beers taste different and they are pasteurized. No, you don't know beer either. All right, doesn't matter. All milk, at least in Oregon and Washington, cannot be sold unless it is pasteurized. I've never tasted unpasteurized milk, but apparently it tastes different, and that's why a lot of people liked unpasteurized milk. The trouble was, if you don't pasteurize it, you can pass on human pathogens. And so the states of Oregon and Washington said that, no, you can't sell or even give away unpasteurized milk. It has to be pasteurized. And it's just a health law of those two states. And I'm sorry, I don't know about the other states. I just don't know. But in Oregon and Washington, all milk has to be pasteurized. Many years ago, and I don't know when that law came into effect, maybe the 1960s or 1950s, people could get unpasteurized milk. Okay, but that was a long time ago.
I've had goat's milk. It's pretty disgusting. Unpasteurized goat's milk, just straight from the boob.
Okay. And is that because you know a goat farmer or you have your own goats?
I grew up on a ranch, so we drank it from there, but it was disgusting. You could taste, like, you'd have to really wash the boob off, the udder, I guess you consider. But it tasted like goat.
Okay. Anyways, even goat's milk, at least in the state of Oregon and Washington, if you're selling it, it has to be pasteurized. Most people don't drink goat's milk, but yeah, that's just a state law for Oregon and Washington. Okay, the germ theory of disease. The concept of fermentation, the realization that microbes can affect an organic material and change it, meaning changing the sugar into alcohol, brought up the question, could microbes have a similar relationship with plants and animals? And could microorganisms cause disease? This idea became known as the germ theory of disease. Originally, poisonous vapors were believed to be responsible for at least some infectious disease. Like people said, the reason why you got cholera was you were walking in the swamp and you breathed in the poisonous vapors of the swamp, and that's why you got cholera. Of course, the real reason why they got cholera was the bacteria causing cholera were in the swamp, and as you were walking through the swamp, you breathed in or consumed some forms of cholera. Okay? Semmelweis advocated hand washing to prevent the transmission of a disease. It's postpartum or puerperal fever. That was a disease a pregnant mother got during childbirth. And he just showed, he actually had evidence that when the doctors washed their hands before giving birth to a child, that the mother would be much less likely to get an infection and get disease. And his idea in 1840 was so radical that he was actually fired because doctors were insulted by the idea that they had to wash their hands before doing anything in the hospital. Joseph Lister, a little while later, used a chemical disinfectant to prevent surgical wounds, and he dramatically reduced surgery-related infections. Before his time, the doctor, when he did surgery, they didn't have any anesthesia. And is that the right word? Yes. And so the patient, if the surgery was successful, the patient was likely to die of the infection in the surgery. And Joseph Lister proved that surgical infections were caused by microbes, and you could prevent it by using disinfection or antisepsis. I should mention that carboxylic acid, a phenolic compound, was what he used, and that is not allowed to be used on patients today because it's actually a carcinogen. But if the patient was only exposed to it one time, it would have, in his day, prevent the infection. And if they're only exposed to it one time, they probably would not have gotten cancer. And that was the best chemical disinfectant he had found. The germ theory of disease was then further developed by Robert Koch, Dr. Koch. And by the way, if you're German, you can pronounce that much better than I can. It's some German sound that is not in English, like something like that. And I just can't say it. I'm sorry, don't speak German, and I can't say that sound. But you can pronounce that as coach or cock, or I don't know. Like I said, in English, the term isn't, the sounds aren't there for making the correct sound. He proved the bacterium Bacillus anthracis causes the disease anthrax, and he provided the experimental steps required to prove that a specific microbe causes a specific disease. And those experimental steps are known as Koch's postulates. And that proved the germ theory of disease because the first germ, Bacillus anthracis, was now shown to cause a human disease, anthrax. And as I stated, he was very lucky and very good about doing this because he was looking at an organism that was big for a prokaryotic cell, so it was easy to view, and the microscopes weren't that good in his day, and the staining really wasn't developed, and so he was able to see him with his simple primitive stains. And then Bacillus anthracis does have some structure to it, and so we could distinguish that bacterium from another, and that was because it had endospores. Anyways, he then wrote up his steps for how he proved that the Bacterium Bacillus anthracis causes anthrax, and those steps are known as Koch's postulates, and you don't need to know the year he published that. All right, I think we're getting close to the time we're supposed to stop. Is that right? Oh no, it goes to 3:45. So let's keep going. Make sure I'm on the right slide. Another thing that happened around this time, it actually happened before these discoveries in microbiology, and that is the first vaccination. The first vaccination happened or occurred because of Edward Jenner, and he inoculated, actually it was 2 boys with cowpox, and that's another virus that causes a slightly different disease than smallpox, and he was trying to protect them from smallpox, and he really didn't have the experimental proof that you would need today to show that cowpox was safe and that it protected from smallpox and all that. He simply had empirical knowledge, and he believed it was strong enough to go ahead and do this. And it was surprising to me today, and maybe to you as well, that he was experimenting on children. Okay, you could never do that today. Of course, smallpox was a dreaded disease back in his day. The parents probably were willing to trust Edward Jenner when he said that I can develop a way to protect your sons from getting smallpox, and they will get cowpox. And cowpox was a really mild disease that actually Edward Jenner had noticed that milkmaidens got. And on the milkmaids, you could see one, I don't know if you can see my thing, someplace on the arm or maybe on the hand, one lesion, which was a pox lesion, that was cowpox. And that's all that the patient who got cowpox got. And that's different from smallpox, where the patient would be covered in these pox lesions. And the cowpox was a very mild disease. The individual might get a headache and maybe even a slight temperature, but in about a day or 2 later, they'd be feeling better. And smallpox, of course, was so bad that it killed people. And I'm not talking about a small percentage. There are different strains of smallpox. The worst strain could kill up to 20% of the patients. The other strains of smallpox killed at least 1 to 2% of the patients. And so it was a lethal disease, and people were probably willing to have their children inoculated with cowpox. And Edward Jenner had no proof that it would succeed until he inoculated the 2 boys. One of them was his own son. And those 2 children did not get smallpox when smallpox went through their area of England. And that was the first vaccine that happened in 1796. There were no other vaccines for about 100 years later. And that's because nobody understood why Edward Jenner's vaccine worked. What he did was he took the pus from a cowpox patient and injected it into an individual, and then that individual, they might get cowpox, which was very mild, but then after that, they were immune to smallpox. But nobody understood why that was happening. And it wasn't understood until the experimental work of Louis Pasteur again, who showed that what was happening was the cowpox virus was related to the smallpox virus, and that when our immune system of the patient mounted an immune response against cowpox, it also mounted an immune response against smallpox. And that protected the patient from getting smallpox. And then after Louis Pasteur's work, other people started developing vaccines to protect against other diseases. And vaccination is actually derived from the word vacca, which means cow, and that comes from cowpox. The first vaccine was using cowpox, so that's where the term vaccination comes from. Any questions about that? The protection the patient gets from a vaccine is called immunity. And that's just your immune response reacting to the vaccine. And hopefully, someday we'll have that to COVID-19, because until that day happens, we're not going to be going back to things as normal. Although we can get closer to normal. And I think Washington is slowly moving that way. Does anyone know anything about when Washington is going to start opening up? I know at Clark College, there are a few classes that are meeting, but those are really classes that you need hands-on learning, like, oh gosh, what is it? Where you're working on the motors and things like that. You can't do that online. And I believe those students have to wear masks, and the rooms have to be not very crowded, meaning the students have to be spaced out. And when you arrive on campus, you have to wear masks and probably gloves, and you go through that. We were talking about maybe having the lab go that way, and maybe someday in the future we'll do that. But for the time being, we're totally online for microbiology lab. All right, any questions about that? I said the state of Washington and the state of Oregon. My apologies, you're in the state of Arizona. Why didn't someone correct me? I don't know what the pasteurization laws are for Arizona. I'll have to look it up. And I don't know what any of the state of Arizona things are. Right, I'm getting a little confused. I'm sorry I teach in two places, so because it's online. All right, any questions there? Gosh, you guys should correct me when I make a mistake like that. No problem, I like corrections, okay? And when I make a blunder like that, I'll give you an extra credit point, okay? All right, what's the time here? Oh, we got a little more time. Out of Louis Pasteur and Koch's work, and really the germ theory of disease, came the birth of modern chemotherapy. And chemotherapy is, strictly speaking, the treatment of disease with chemicals, but nowadays, in common usage, chemotherapy is used for a term used for treating cancer. And so we won't use that term for the treatment of disease with chemicals. We will use either chemotherapeutic agent to treat an infectious disease, or just generally the term antibiotic. Yeah, good. I don't have that shown here. So the concept is that we could find a chemical that would affect the organism causing the disease, but leave the patient mostly or totally unharmed. And that is where the concept of chemotherapy came out with. And the chemotherapeutic drugs can be a synthetic drug, that's one that's man-made, or an antibiotic, which is the textbook definition of the term antibiotic, and that is a chemical produced by a living organism, either a bacteria or a fungi, that inhibits or kills other microorganisms. And as I stated, that term of an antibiotic is the textbook definition. And you can have something like a semi-synthetic, which is made by a microorganism and then chemically modified, but it's not a synthetic drug where the humans totally make it 100%. As we will use the term antibiotics will be the way it's used in common usage in the population, and the term you'll find in most clinics, I'm not going to say all clinics, but most doctors will use the term antibiotic as any drug that inhibits or kills a microorganism. And if you think about it, if you're talking about carboxycillin, which is a synthetic drug, so you can't call it an antibiotic because it's not made by a living organism, but then penicillin G, which is in the same family of drugs, is made by a microorganism, so we call it an antibiotic. That doesn't make a whole lot of sense to separate those two because they're in the same family of drugs. Okay, so we'll use the term antibiotic as it is commonly used. The first chemotherapeutic agent that was used for treating an infectious disease was quinine from the bark of a tree, the cinchona tree, which you don't need to know. It was the first drug widely available in Western Europe that was used to treat a disease. It was used to treat malaria. And would anybody like to guess when that first chemotherapeutic agent was discovered and written about in Western Europe? Just take a guess on when you think that was.
Early
The 1930s?
The 1930s, and Christopher, you're going to say something about the same?
I was going to say early 1900s.
Early 1900s, okay. That would be sense. That would make sense because that's when Paul Ehrlich was actively looking for a chemotherapeutic agent. But no, you're actually wrong. The first chemotherapeutic agent was discovered or was written about in Europe in the mid-1600s. Jesuit missionaries in Peru noticed the natives eating the bark of the cinchona tree. You don't need to know the name of the tree. And it had quinine in the bark, and they used it to treat their malaria. And the Jesuit missionary said, hey, that works. Let's take the bark home to Europe and use it to treat malaria in Europe. And that's what they did. So way back in the 1600s is when we had our first chemotherapeutic agent. The next chemotherapeutic agent was not found again until 1910. And it was found by Paul Ehrlich, who was looking for the magic bullet. I'm doing quotes when I do that, okay? He was proposing that we could find a chemical that would cure and kill, or kill and cure, a disease-causing organism in a patient and leave the patient pretty much unharmed. And he just went chemical after chemical, testing it in rabbits, one after the other. And after about testing 500 chemicals, he came across one that was successful in treating the disease syphilis. Now, it was used in 1910, but it was a synthetic arsenic drug, salvarsarin, which, as you can imagine, you don't generally want to take arsenic. Okay? So yes, it did cure malaria, but it had pretty harsh side effects, meaning the patient was pretty sick from taking the drug itself. Any questions about that? All right. And I think we'll stop here. It's getting close enough to the stopping time, and we will stop on the first antibiotic. Any questions? If not, I will see you tomorrow.
I have something.
Okay.
How detailed is the history going to be on quizzes and the final?
There will
Like, I understand like the order of everything, but like how detailed of every instance.
You don't need to know the date, but on many of the other items, you need to know what we discussed. Like you should know quinine was the first drug used to treat malaria. You don't need to know the date that that was used. You should know that Edward Jenner came up with the first vaccine which protected against smallpox, and it was using cowpox. So is that the detail you're asking?
Yeah, sort of. So pretty much what's written in these PowerPoints and not so much the expanded version that you're speaking.
Okay.
Sort of. Sort of.
Sort
Yeah.
of. Okay.
Okay, just checking.
All right. All right, I'll see you tomorrow.